Welcome to Ask the Expert. It's a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event. We're going to post it on the Sugar Science site YouTube channel shortly after presentation. If you have questions for our guests, feel free to enter them in the chat or raise your hand at the end of the presentation. And today we're so lucky and excited to have as our guest, Dr. Megan Penno at the University of Adelaide in Australia. And Dr. Penno is the National Project Manager for ENDIA, which is the Environmental Determinants of Islet Autoimmunity Study. She's also a research fellow at the University of Adelaide, Australia. ENDIA is the first study in the world to explore how environmental exposures from pregnancy through early life may contribute to or protect against the development of childhood type 1 diabetes. Megan was also appointed to manage ENDIA at the project's inception in 2012. And she's played a central role in establishing and expanding the cohort, which completed recruitment of 1,500 mother-baby pairs in December 2019. Drawing on her experience in omics research, Megan was the first author of the published ENDIA protocol in 2013, and she's developed the sample and data collection strategies that will underpin all future investigations involving ENDIA participants. And she's now leading her own research project within the ENDIA study, aimed at identifying changes in the plasma proteins of mothers during pregnancy and in babies during early life that may indicate a child is at risk for developing type 1 diabetes even before the destructive autoimmune process has commenced. This work is funded by JDRF Australia, the Leona M. and Harry B. Helmsley Charitable Trust, and Diabetes South Australia. Welcome, Megan. Thank you so much for joining us.
Thank you, Monica, and thank you to the Sugar Science for having me on today. It's a real honour to be able to share with you the research that we're doing all the way down here in Australia. And I think Monica asked me before we get started on the science to just share a little bit about myself and my journey.
I love this progression. Fantastic.
Yeah, so basically, I grew up in country Victoria and went to university at the University of Melbourne and did my PhD at the Australian Animal Health Laboratory, part of the CSIRO network in Geelong between 2002 to 2006. And my interest there was actually looking at sheep. I was doing a biomarker discovery project looking at sheep proteins, trying to identify markers of toxin exposure. So on completion of my PhD, I moved to Adelaide and I stayed in the field of proteomics, transitioning to more human-based studies and in particular a biomarker discovery project for gastric cancer. The proteomics lab that I worked in at the University of Adelaide also did a lot of fee-for-service type research because proteomic platform technologies are pretty expensive. So it was here that I first encountered samples that were collected as part of clinical research trials. And I think my experience there was not always great. So sometimes we would end up with tubes that would have like 52 written on the lid, and somehow I was supposed to take this tube and make some sort of biological sense out of the contents of that tube. And that was really difficult because the work we were doing, it was really precise and really expensive, but we didn't always know that the quality of the sample that we were receiving was going to deliver the results that the researchers were after. So with that sort of practical experience, in 2012, I came across an opportunity to get involved in human research as a project manager of a national type 1 diabetes study. And I thought with my, skills and background and experience in the lab, I could have something to offer in terms of, designing the study and putting it together and making sure the labels didn't fall off the tubes when they came to the downstream researchers. And so I was fortunate enough to get that position in project management. And that's where I've been since 2012. So whilst I've been out of the laboratory for quite a number of years, I'm actually coming full circle now. And as you mentioned, Monica, are now leading a sort of proteomics biomarker discovery project using the samples that we've been collecting for the past 8 years. So these are sort of my photos. I promise my hair has changed a little bit more. than these photos show. And of course, the most recent one taken just the other week down in Melbourne, COVID-19 has certainly changed our lives. And I think this photo shows that.
Yes.
So let me move on to, I guess, the real hero of the show, which is type 1 diabetes research. And just to, I guess, set the scene in an Australian context, as of June, the end of June this year, there are approximately 127,000 people in Australia who are living with type 1 diabetes. And each day there are 4 children and 3 adults who are given a diagnosis of type 1 diabetes in Australia, which is 7 people too many.
Yeah.
Like in many parts of the world, the incidence of type 1 diabetes in Australia is increasing, having doubled over the past sort of 4 decades. So in the 1980s, per 100,000 children aged 0 to 14 years, 11 children were diagnosed with type 1 diabetes. Fast forward to the most recent data in 2018, and we have 24 children per 100,000 who are diagnosed each year with type 1 diabetes. And as you can see from this graph here, the incidence fairly closely resembles the incidence that we see in Colorado.
Yeah.
So in parallel to this increasing incidence of type 1 diabetes that we've sort of seen over the past 30 to 40 years, paediatricians have also seen a rise in other disorders that affect children, including overweight and obesity, allergic diseases, autism spectrum disorder, and attention deficit hyperactivity disorders, inflammatory bowel disease, celiac disease, which interestingly has a genetic link to type 1 diabetes, as well as type 2 diabetes. So just coming back to type 1 diabetes, the relative frequency of what we consider to be high-risk genotypes amongst newly diagnosed children has been steadily decreasing since the 1950s. So this suggests that factors external to the genome, so the environment and perhaps more specifically the modern environment, are driving this increased incidence of type 1 diabetes that we're seeing worldwide.
Yeah.
So there is fairly substantial evidence to indicate that the modern environmental exposures that are driving the development of type 1 diabetes are being encountered very early in childhood, and perhaps even during pregnancy and potentially even preconception. And many of these exposures are sort of summarised in this graphic here. Indeed, the earliest evidence that we currently have that a person has commenced on a trajectory towards type 1 diabetes is via the presence of islet autoantibodies in the bloodstream, which we frequently see within the first years of life. So here we've got 2 graphs. The top one represents the age at which children participating in the TEDdy study have developed islet autoimmunity. So among these almost 550 children that were participating in this study, the incidence of islet autoimmunity peaked at 9 months of age. Data obtained from our own India population, although currently much smaller than the TEDdy population, is showing the same thing, with most of the India children developing the islet autoantibodies within the first year of life. So whilst there are lots of fingers that are being pointed at sort of prenatal exposures being relevant in the natural history of type 1 diabetes, before 2013, there were no type 1 diabetes risk cohorts with prospective data collection that was commencing in pregnancy anywhere in the world.
So Megan, would you just say that this, with India and sort of TEDdy there, you guys are establishing a window of, you know, I guess etiology of type 1, because we know it's very heterogeneous, right? There can be different time points of diagnosis, but so you're establishing, it seems to me like you're establishing a 9-month, the window for a 9-month diagnosis. Is that right?
Yeah, so I think it kind of comes back to this concept of like this developmental origins of health and disease, or DOHAD hypothesis. So this hypothesis is based on the fact that exposures during the pregnancy and very early in life, and even perhaps pre-pregnancy, kind of set up, you know, the biology that affects you for the rest of your life, and also potentially what you're passing on to future generations. So whilst you're right. I mean, type 1 diabetes is very heterogeneous in terms of its age of onset. And I did mention that, in Australia, 4 children, but, 3 adults every day are diagnosed with type 1 diabetes. But I think what we see from the islet autoantibody work is that a majority of people who are diagnosed with type 1 diabetes, in childhood, even up to the age of sort of 18 years of age, have got these antibodies when they're very young. Yeah. So the sort of precipitating event, it may take 10 years or 20 years to manifest, but it's potentially an event that occurred very early in life. So we're interested in this period, I guess, looking at early onset type 1 diabetes in very young children, but it may be relevant to the life course and type 1 diabetes.
Yeah, the prodrome can extend. But yeah, correct. I just wanted to kind of just frame that. Okay, great. So let's dive into the India study.
Yeah. So yeah, I mean, to address the kind of this DOHAT hypothesis around the origins of type 1 diabetes, a group of pediatric endocrinologists right across Australia established our India study. And the central aim of this study was to identify the environmental exposures and those gene environmental interactions from the pregnancy that triggers islet autoimmunity, which eventually leads to type 1 diabetes. So this study was set up across 9 clinical sites in 5 Australian states, and also incorporates a collaborative network that includes 8 of Australia's major universities, including the University of Adelaide, which is responsible for the administration of the India study, as well as 6 medical research institutes. So you can see on this map of Australia, there are lots of little blue dots, and those blue dots represent the locations of where our India participants live. So there's blue dots that are quite a long way away from some of our clinical sites. And that's because we have a regional program as part of the India study that enables eligible families who live anywhere in Australia to participate in this research. So the eligibility criteria for India were an unborn child recruited during the pregnancy, or a child less than 6 months of age who have an immediate family member. So in other words, a mother, a father, or a sibling who had already been diagnosed with type 1 diabetes. And the reason why we were recruiting children who have a first-degree relative was because these children are at increased risk of developing type 1 diabetes. Their risk is around 16 times higher than if we were to recruit children from the general population. So as I mentioned, recruitment commenced in 2013, and I'm very pleased to say it was completed in 2019. And our very last baby was born, clearly by this photo, as you can see, in the middle of the COVID-19 pandemic in July 2020. So I guess a rather sort of scientifically dry but sound way of looking at recruitment can be summarised here in this strobe diagram, which shows that of all of the people who were approached to participate in India, 1,511, or about 55%, consented to participate. So 12 of those who consented didn't actually commence, and then a further 4 were found to be ineligible subsequent to starting, resulting in an eligible commenced population of 1495. So very sadly, when dealing with a pregnancy cohort, we did have 22 adverse pregnancy and neonatal outcomes within our families. And we would really wish to acknowledge those lives and really thank those families for their willingness to be involved in our research. So when we take it all together, the India study now consists of a population of 1,473 infants who were born to 1,453 pregnancies that were carried by 1,213 unique mothers. So this is the dry way of looking at it. The way we prefer to look at it is like this. And we are so lucky because our India families continue to send us really beautiful photographs and share their lives with us, which we're able to put on social media and include in our presentations. So as you can see here, we did have more babies than pregnancies. And that's because our cohort included 18 sets of twins and 1 set of triplets. And there were more pregnancies than there were families because 220 of our families participated with more than one child who was recruited who was born, during the recruitment period. In terms of what the India cohort looks like, the age gap between our youngest and our oldest India child, so this is our oldest boy and this is our youngest girl, is 7.6 years. Presently, the median age of our cohort is 3.9 years. So of our 1473 India babies, a combined 62% have a mother with type 1 diabetes, 28% have a father with type 1 diabetes, 12% have a sibling, and there are 3% of children that have multiple family members, multiple first-degree family members who are living with type 1. So this cartoon here depicts what the follow-up in the India study looks like. So visits are occurring 3 monthly from the pregnancy until 2 years of age and 6 monthly thereafter. So the follow-up in India can be sort of broadly divided into 4 different categories. So we're interested in health events, and this includes things like growth and illnesses, medication usage and mental health. Secondly, we're interested in lifestyle and physical environmental factors. So this is things like family structure, physical activity and pet ownership, that type of thing. Thirdly, we're interested in nutrition in both the mother and in the baby. And lastly, biospecimen collection is a big part of India, and this is what I'm particularly interested in. So here we have another diagram that depicts all of the different types of biospecimens that we collect and the timing of those collections. And it's important to note that we take samples not just from the mothers and the babies, but also from the fathers and the other family members too. So basically our motto in the India study is if it leaks out of a person, we scoop it into a tube and put it in the freezer.
That's great.
So from across the pregnancy until 10 years of age is the point that we're following these children. India collects around 200 unique biospecimens from each family. So with our 1473 families, this equates to almost 300,000 samples from our whole cohort. And if you think about the fact that those 300,000 samples are split on average into 5 different aliquots when we store them, we anticipate that the India study will have around 1.5 million individual tubes stored in freezers around the place across Australia. So we need a lot of freezers.
Yeah, a lot of freezers, and then they all need analysis.
Oh, exactly.
That'll be the bigger hill to climb, don't you think?
Oh, exactly. And then there's the data, but we'll get to that later.
Okay, great.
Yeah, so as I mentioned, all the children in India do have an increased risk of developing type 1 diabetes because they have this first-degree relative with type 1, and that the first signs that a child is progressing towards the development of type 1 diabetes is via the detection of these islet autoantibodies. So islet autoantibody, islet autoimmunity is the primary outcome of the India study. And as this table up here shows, that children who have 2 or more of these islet autoantibodies have quite a high risk of progressing to type 1 diabetes by 15 years of age. So based on our 1473 children that we have enrolled, we project that somewhere between 6 and 8%, or approximately 100 children, will develop islet autoimmunity by the end of 2024. So at the moment we're sitting here at 62, and we appear to be tracking fairly closely towards our projected figures, although COVID has caused a few problems with challenges in obtaining those blood tests. But yes, as I mentioned, 62 children so far have developed persistent islet autoimmunity, and 15 of those have already progressed to type 1 diabetes. So at the end of 2019, there were 54 children who developed persistent islet autoimmunity, which represented the approximate halfway mark of the India study. So at this point, the team decided to commence sort of these first exploratory investigations, looking at associations between various exposures, and in particular omics measures in the pregnancy and early life, and the development of islet autoimmunity is the outcome. And so this study design, a nested case control study, was established by the team of biostatisticians at the University of Adelaide. So the cases in our nested case control study, and again, you'll see another strobe diagram here for the NCC with 1473 participants at the top. So the cases are the first 54 children who developed islet autoimmunity up until the end of 2019. And these 54 children have been matched based on their date of birth and sex to 161 other children also participating in India who were antibody negative at the time that that case was first found to have the islet auto antibodies. So 53 of the cases were matched to 3 controls each, and there was one child that had only 2 eligible age and sex match controls. So this gave us that population of 161 controls. Now, because in this nested case control design, control sampling occurred with replacement, that means that individuals could be matched multiple times to multiple different controls. So our final NCC design comprised 190 unique mother-infant dyads. And our plan is to undertake various omics studies using these same 190 dyads across different omics platforms. So this diagram here, I guess, represents the suite of the different types of omics investigations that we're planning to undertake using our NCC design. And I'm pleased to say that most of these investigations, in fact, are funded. So up here, the work on the bacterial microbiome and fungal microbiome from various body sites will be performed by our collaborators at WeHi in Melbourne. The virome studies of the gut and respiratory virome, as well as viral serology, will be undertaken at the University of New South Wales in Sydney in collaboration with Columbia University in New York. The proteomics work, looking at the plasma proteome, will be driven by the University of Adelaide, by myself, in collaboration with WeHi in Melbourne. While we actually have another group who are looking at the gut metaproteome, which is largely the proteins that are produced by the bacteria living in the gut, and that will be undertaken by collaborators at the University of Queensland. Lipidomic studies on the plasma will also be done in collaboration with the Baker Institute in Melbourne. And we have some international collaborators who are interested in looking at the plasma glycome, so the glycoprotein component of those plasma proteins. The basic genotyping efforts for HLA imputation are currently underway at the Harry Perkins Institute in Perth, but we do have plans to expand this work to include high throughput genotyping arrays. And in addition to this, we're also looking at epigenetics and gene expression in specific subsets of immune cells happening at the University of Adelaide and at WeHi. And of course, underpinning all of these omics investigations, we also have the near 7,000 fields of clinical data that are collected from each of our participants, which span things like nutrition and growth and most lifestyle factors.
Sorry, I just was going to jump in there and say this is a tremendous team sport. It's really admirable. I wondered, you know, for the bacterial, we're kind of really interested in this sort of bacterial microbiome here and the stool metaproteome. Those two, like, what's the timeline for discovery of those two? Do you just sort of a guesstimate?
As in, like, when are we commencing this type of work?
Yeah,
or-
when is it commencing, and what
Yeah.
kind of a timeline is out there for sort of, discovery, I guess?
Until we see these results, yeah.
Yeah.
So actually, as I mentioned, we're really fortunate that most of these studies that are shown on the slide are indeed funded, and the bacterial microbiome work is well underway. So the whole metagenomic sequencing that's being performed on these samples is complete for about half of the participants that are involved in this cohort.
Oh, wow. That's
There's still metaproteome work, which is being done, led by Emma Hamilton-Williams at the University of Queensland. So she's just starting to undertake those investigations now. So in reality, it'll probably take us at least the rest of this year, leading into next year, to acquire those data. And then I think, as we alluded to earlier, the huge challenge is sort of in the analysis of these complicated big data sets. So it would be great to think that, we would see the outcomes of these studies sort of published moving into 2023.
okay, great. Yeah. And who's the head at the WEHI?
At WEHI? Yeah, so that's Professor Len Harrison who's leading that work then.
Interesting. Okay. So if anyone's interested in that type of work, those are the Emma Hamilton-Williams and Len Harrison are leading the charge on those. I know we have a lot of interest from the audience, about those particular targets.
Fantastic. Okay, so I mean, we're talking about size here, right? And in addition to the large number of omics platforms that we'll be using to analyze these samples, it's also important to remember that these samples are collected longitudinally in India. So for each of the omics studies that we showed on the previous slide, we plan to look at all of the samples that have been collected from these mother-infant dyads from the pregnancy at birth and up until the point of development of islet autoimmunity in this case. So in this figure here, each of these tiny dots, and there's 1,000 of them, represents a plasma sample that will be included in the plasma proteomics investigations. And the little teeny tiny red dots that you can see here represent the time point of the onset of islet autoimmunity in the cases. So you can sort of see that the youngest person to develop islet autoimmunity in our cohort was 4 months of age, and the oldest child that we have was 5 years of age. So this is just sort of the plasma dots. For the other sample types that we've collected, the sample numbers range from 500, just over 500 samples of breast milk, through to nearly 50 or more than 1,500 throat swabs. So when we look at these multiple samples using multiple different omics platform technologies, that represents several terabytes of big data that will be generated by this project. And of course, bringing all that together will be a fairly substantial task. So the results so far, I mean, this comes back to your question, Monica. Basically, we don't have any for our NCC cohort yet. But what we really have been working on for the past 8 years, I suppose, apart from acquiring enormous amounts of data and enormous numbers of biological specimens, we've been looking at kind of acquiring what we're kind of calling normative data, comparing the type 1 versus the non-type 1 pregnancy, looking at the gut virome, the bacterial microbiome, and also the fungal microbiome. And we've also done some preliminary studies outside of this nested case control design, looking at exocrine pancreas function with islet autoimmunity as an outcome. So I'll just give you a quick 1-slide summary of each of these. So regarding the virome, and this work has been led by colleagues at the University of New South Wales in Sydney. So Ki-Wook Kim, Maria Craig, and Bill Rawlinson. Again, names to look up if you're interested. And these folks have been using a technique called VCAPSEC, which was developed by Columbia University. And VCAPSEC is capable of measuring 10,000 different virus species that affect vertebrates in a single sample. So it really is an omics technology for looking at viruses. And so for the first time, what they did was compare mothers with and without type 1 diabetes in pregnancy across the 3 trimesters to look at the populations of viruses that were present in these women. And what they found was distinguishable characteristics between the women with and without type 1 diabetes, including a higher prevalence of picobeanoviruses and tabamoviruses in the women with type 1 diabetes, while the viral load or the abundance of enterovirus B, which is particularly interesting for type 1 diabetes research, was higher in the mothers with type 1. They also looked at 24 babies who were born to a subset of these mothers. And they were able to find evidence of viral infection in all children except one, which would be no surprise to anyone that's ever seen a child. And in total, they were able to find 26 different genera of viruses. And again, they were able to identify distinct patterns that separated infants born to mothers with type 1 diabetes versus infants born to mothers without. But interestingly, those viruses in these children were not quite the same as the viruses that were found in the mothers. So at present, we actually don't know what the differences mean and whether this has any impact on a child's risk of developing type 1 diabetes. But what we do know is that children who are born to a mother with type 1 diabetes actually have a lower risk of developing the condition themselves than children who are born to a father with type 1 diabetes. So we don't know whether some of these differences that exist in the virome may play a role in protecting child from development of type 1 diabetes, and this will be further explored in our NCC study. So coming to the microbiome and the mycobiome, so the analysis of the bacterial microbiome by whole metagenomic sequencing, which incidentally was just published this week in Microbiome, which is pretty exciting.
Congratulations, that's great.
Yeah, no, that paper had a gestation that almost felt like an elephant, but anyway, we got there in the end, and it's really fantastic to see that work. So yeah, so we looked at the bacterial microbiome by whole metagenomic sequencing. We've also been looking at the mycobiome by ITS1 amplicon sequencing, and there's a manuscript associated with that work that is currently under review. And these work taken together have revealed some important characteristics also of the type 1 versus a non-type 1 pregnancy. So what we've shown is that although the microbiome does change across pregnancy in all women, it does so distinctly in women who have type 1 diabetes, and particularly in the 3rd trimester. And that's evidenced by this figure here, that shows that a particular species, so this is Bacteroides uniformis, it increases in abundance with type 1 in women with type 1 diabetes across pregnancy, but decreases across pregnancy in women without type 1 diabetes. So the combined results of our microbiome and mycobiome studies have also found that pregnant women with type 1 diabetes exhibit a decrease in bacteria that produce short-chain fatty acids and B group vitamins, while they actually show an increase in LPS-producing bacteria and a higher abundance of the fungus Saccharomyces cerevisiae. At the same time, we've shown that 3 different markers of gut pathology, that being fecal calprotectin, and the serum levels of intestinal fatty acid binding protein, as well as antibodies that target Saccharomyces, are increased in women with type 1 diabetes. So taken together, I think the gut microbiome, the mycobiome, and this combination of impaired intestinal barrier function may be contributing to the maternal and the fetal complications that are more prevalent in type 1 diabetes. And this is a really important finding because, if we can implicate the microbiome, it gives us opportunities to potentially intervene, which could improve the outcomes for mothers and babies who are born to women with type 1 diabetes. So this is really quite important work on itself.
Yeah, this is huge, really. I mean, it just shows a true signature of a snapshot of the type 1 diabetic pregnancy.
Yeah, yeah, exactly. And it's an area. I mean, pregnancy in general. There's not a lot of out there, particularly in the microbiome, which is surprising. We're all resulting from a pregnancy, but it's a really underappreciated area of research, and particularly type 1. One of the things that I've been struck with working in the India study is the rate of complications that women who are experiencing a pregnancy living with type 1 diabetes go through. And I think that, if our research can deliver on work that will improve the outcomes for these women in their pregnancy, in addition to perhaps what we can do looking at the etiology of type 1 diabetes, but just improving that type 1 pregnancy is something that we're really passionate about.
Yeah,
and
it
have
has
an opportunity to do.
well, it has direct application to the clinic.
Absolutely, absolutely. Just the sort of last piece of work here that I wanted to share with you is some work that was led by the University of Adelaide, and we published this last year in Pediatric Diabetes, and it involved the measurement of a protein called fecal elastase in the stool samples from 28 children. Our first 28 children that developed islet autoimmunity, and we compared this with 57 matched children who were antibody negative. And this protein fecal elastase is a surrogate marker for exocrine pancreas function. Mm-hmm. And what we found that the fecal elastase levels did not differ between sort of the antibody positive and the antibody negative children at baseline. And this suggests that children who develop islet autoimmunity actually didn't have a smaller pancreas from birth. So that kind of wasn't the issue here. Regression modeling performed by our statisticians showed that the trajectories or the slopes of these fecal elastase levels in the control groups were increasing as the children aged, but in our case groups were decreasing. And importantly, in some of our cases, this fall in fecal elastase actually occurred prior to the onset of islet autoimmunity. And although the values of fecal elastase were still considered within the normal reference ranges, these levels were falling, but they were considered within the normal reference ranges. And we actually sort of did see a high amount of variability even amongst the same children. So the conclusion that we can draw from this piece of work that, whilst we can implicate the exocrine pancreas in type 1 diabetes pathogenesis, which does support the concept that type 1 diabetes is a disease of the whole pancreas and not just the endocrine component. Yeah. However, using fecal elastase in itself as a biomarker for sort of predicting or stratifying type 1 diabetes risk is not particularly useful because of this heterogeneity that we see. So hopefully some of our other omics investigations that I talked about will lead to the identification of improved biomarkers that we can use for risk stratification. And this is particularly important for potential entry points into future intervention studies where we may be interested in primary but also in secondary prevention.
Yeah, no, it's very interesting to see what's happening with and there's a lot of sort of fingers pointing at the exocrine pancreas, that the alpha is also involved. And so this is a great way to look at that, I think.
Yeah. And so this is kind of also quite preliminary work. It was done based on a relatively small number of cases. So we hope to be able to expand this in the future as we see more islet autoimmunity appearing in the India cohort.
Yeah, first steps, but yeah, it has a lot of promise.
Yeah. And really, with that, I mean, that's sort of the conclusion of my presentation today. I would really like to acknowledge all of the fantastic people that are involved in India. We, like our little children that we've been following, we've really grown over the years. So this series of photos just shows our study team meeting from 2016, 17, 18, 19, And of course, this is the 2021 photo where we're unable to meet in person. We're all on Zoom, but we do the best we can. There are too many people to name, but I'd particularly like to acknowledge the principal investigator of NDIA, Professor Jenny Cooper, based at the University of Adelaide. I'd of course also like to thank all of the beautiful families that we have the privilege of following. So this is a bit of a montage of many of the images, and really, without the dedication of those families, we wouldn't have the study that we have today. So sincere thanks to all our families. And of course, I'd like to acknowledge all of the participating institutes, of which there are many, and particularly our funders, JDRF Australia and the Helmsley Charitable Trust. We're also funded by the NHMRC in Australia and Diabetes South Australia, me personally. So thank you, Monica, and thank you everyone for your attention. And if any questions, I'm happy to answer.
Yeah, I mean, if anyone has a question, just if you don't need to, you can unmute yourself, or you can just drop it in the chat. I'd also like to just say, I think that last slide where you showed, definitely all the participants, but all the scientists that are involved, I think it's a real, it has such an important message, I think, to those who actually have type 1, that scientists are working so hard on this problem. And I think sometimes out there in the actual struggle, day-to-day struggle for parents to try to keep their little ones, especially the little kids, on target with their blood glucose and all these other challenges that come with type 1 diabetes, it's important for them to know that scientists are, really meeting this head on, and with the most powerful tools that are available, as well as, just really going after big amounts of data now to try to get to the bottom of this. So I think that was great that you showed that, and thank you.
My pleasure. Yeah, I mean, we really see, and we've discussed within the team, there's sort of almost a passing of the baton from, we've followed these families. Our coordinators across the country have been absolutely brilliant in dealing with our families, collecting all of these samples. We've got a freezer full now, and so that responsibility is kind of being passed on to those scientists in the lab to take this resource and work their magic with it, as it was, and we take that responsibility really seriously, because every blood sample is a venipuncture that a small child had to go through. So we look at these samples with respect, and and look forward to doing our best with them.
I also think there's huge opportunity, for young scientists who are just coming out of graduate school, or as just ending their postdoc, to really explore the interface here with NDIA and labs that are, coordinating with NDIA to, jump on board some of these laboratories and roll their sleeves up, get involved in this data analytics sort of quest. I think this is a really exciting project.
Yeah. And look, the data analysis is a huge challenge. And so if anyone's interested, wants to help, let us know. If you've got some supercomputers, let us know. Because these are big tasks in the integration of these omics data sets. But we look forward to tackling it.
Are you considering interfacing with any omics analytics in industry, or are you maintaining this as a completely academic project?
Look, at present, this is being run as an academic project. So each of the kind of groups that are involved in the acquisition of these data sets are considering how they can interrogate them best themselves. But I mean, that's certainly something that we would be open to. And for the next sort of 12 to 18 months. The data management is something that we're really going to be focusing on. So that's a really great point. I'll look into it. Thanks, Monica.
Yeah, sure. No, I mean, we follow Finch Therapeutics out of Boston, and Ginkgo Bioworks, and companies like that. They're heavily involved in sort of dissecting and understanding the microbiome. There's also in silico medicine, which has a lot of analytics and omics capacity. So we're talking to these companies just to sort of connect them to KOLs and to opportunities. So yeah, maybe,
Cool.
who knows what could come of it. So, but thank you again, Megan. This was just fascinating. I thank you for your time and all the work that is getting done in Australia. A lot of really interesting work coming from Australia these days in type 1 diabetes. So thank you all again.
Thank you so much, Monica, and have a great day.
You too. Bye-bye.
Bye-bye.